Hello everyone. So this is the zoology discussion for NEET mock test 10 and the question starts from 136. So the first statement is PCR is used to detect mutations in genes in suspected cancer patients. Yes, it is correct. A direct method to insert a foreign gene into plant cells is microinjection. No, this is wrong. For plant cells, we use gene gun. So this statement is wrong. The first transgenic cow, Rosie, produced human protein enriched milk that is 5.4 grams per liter. No, this is wrong. It is 2.4 grams per liter. Next statement is RNAi takes place in all eukaryotic organisms as a method of cellular defense. This is also correct. So here the question is asked is how many of the above statements are correct? That is two. Next question. Identify the incorrectly matched pair. Let's see. Humulin goes with first therapeutic rDNA product approved for human use. This is correct. Next, RNAi, that is silencing of mRNA with the help of double-stranded RNA. This is also correct. Rosie, transgenic sheep. No, this is wrong. Producing alpha 1 antitrypsin. This is also wrong. So here, the incorrectly matched pair is option C. Next question is a permanent cure for ADA deficiency could be, and here the straight away the answer is option D, introduction of gene isolated from marrow cells producing ADA. Important is that into early embryonic cells. 139. Polymerase chain reaction is not used in, let's see, confirming presence of a pathogen during early infection. No. Identifying many genetic disorders. This is also in this also PCR is used. Isolating the gene of interest from host DNA to be cloned by recombinant procedure. No, it is not any isolation technique. So the correct answer is C. 140. Match the following. So here patent on basmati rice by an American company goes to the year 1997. First clinical gene therapy goes with 1990. Humulin by Ali Lilly Company goes to 1983. And then construction of first recombinant DNA goes with 1972. So here the correct answer is A. Next question. Insulin used for diabetes was earlier from the pancreas of slaughtered cattle and pigs, but now it is used. No, it is not used now. So this statement is wrong. Reason is that Aarti with some other patients took insulin injections in which insulin was from an animal source and they manifested an allergic response to that. Okay, this reason as a statement is true. So here the correct answer is D. A is false, but R is true. To test the safety of polio vaccine, the organisms used are transgenic mice. 95% mice are used. DNA or RNA segment tagged with a radioactive molecule is called, straight away, the answer is probe. The trigger for activation of toxin of Bacillus thuringiensis is alkaline pH of gut. Again, assertion reason. In 1983, Eli Lilly used E. coli as homologous host for the production of humulin. No, it was an heterologous host. Heterologous. Next, the C peptide is present in mature insulin. No, this is also wrong. So the correct answer is B. Both A and B are false. Bt toxin protein that can kill many insects doesn't kill the bacillus because Bt toxin requires acidic pH for activation. No. Bacillus has protective mechanism against it. No. Then in Bacillus, it is present in an inactive state, and once insects ingest it, it is converted to active form. This is true. So here the correct answer is C. Next, the Indian government has set up GEAC, that is Genetic Engineering Approval Committee, which will make decisions regarding the validity of genetically modified research. Yeah, GM research. Correct. Biopiracy is the term used to refer to the use of bioresources by the native people. No. So here the correct answer is that the statement one is true, but statement two is false. Then match the following. Genetically engineered human insulin will go with E. coli. GM cotton will go with Bacillus thuringiensis. ADA deficiency will go with gene therapy, and the LSR will go with antigen antibody interaction. So here the correct option is D. Which one of the following techniques does not serve the purpose of early diagnosis of a disease for its early treatment? Does not serve. ELISA does. Recombinant DNA technology does. Serum and urine analysis doesn't. PCR technique also it does. Early detection is possible via PCR also. But via serum and urine analysis, it is not possible. They are the conventional uh, methods. Next, true and false. C peptide is not present in mature insulin. True. The insulin produced by our DNA technology has C peptide. False. The proinsulin has C peptide. Of course. True. A peptide and B peptide of insulin are interconnected by disulfide bridges. True. So here the correct answer is A. True, false, true, true. Genetically engineered insulin for human is produced from which of the following? So here the straight away answer is E. coli, that is Escherichia coli. The application of biotechnology includes therapeutic processed food, correct. Diagnostic bioremediation is also correct. Genetically modified crops, energy production also is correct. So all of these. Next, the recombinant DNA technology have made immense impact in the area of healthcare by enabling mass production of safe and more effective therapeutic drugs. As a statement, it is correct, but it is the reason that the recombinant therapeutics do not induce unwanted immunological responses, as is common in case of similar products isolated from non-human resources. As a statement, this statement is also correct, but it is not able to explain the assertion C. So here the correct answer is B. That okay, here that the question arises is that why it is being produced in a mass manner and for the safety and for the safer and more effective therapeutic drugs. Why? So the reason is not being explained here. In RNAi, the genes are silenced using double-stranded RNA. Which part of the tobacco plant is infected by Meloidogyne incognita? It is the roots of the tobacco plant. Golden rice is a promising transgenic crop. When released for cultivation, it will help in. Okay, so golden rice is known for vitamin A. All right, so it is the option is A. Alleviation means to lesser it down. Assertion. Clone of cells having the mutated gene doesn't appear on photographic film in autoradiography. It is correct statement. Next, the used probes do not have complementarity with the mutated gene. This statement, the reason as a statement is also correct. And it is also explaining that why the mutated gene does not appear on the photographic film. Because the probes do not have the complementarity with the mutated gene. So here it is the correct explanation also. Next, how many recombinant therapeutics are being marketed currently in India? The answer is 12. We can grow meristem in vitro to get virus-free plants, correct. Apical and lateral meristems are free of viruses. Apical is free of viruses, but lateral meristem is not. So here the second, yeah, the reason statement is wrong. So here the correct answer is C. Alpha-1 antitrypsin is used to treat emphysema. How many amendment bills have been done to the Indian Patents Bill? It is 2. Transgenic animals can be specifically designed to allow the study of how genes are regulated. Yes, true. true statement. Gene regulation can be studied only in the transgenic animals. No. In the non-transgenic animals also, the gene regulation can be studied. So this statement is wrong. So here, the correct answer is A. Statement 1 is true, but the statement 2 is false. Match the following. Bacillus thuringiensis will go with the cry proteins. Thermus aquaticus will go with the DNA polymerase. Agrobacterium tumefaciens will go with the cloning vector. And Salmonella typhimurium will go with construction of first rDNA molecule. So here the correct answer is A. From the list of human diseases given below, which ones have transgenic models existing for them? For Alzheimer's, it's there. For rheumatoid arthritis, also cystic fibrosis, also in cancer. So nearly for most of the diseases, transgenic models of the animals are present. Next, true and false. The recombinant DNA technology have made immense impact in the area of healthcare by enabling mass production of safe and more effective therapeutic drugs. It is a true statement, as we have seen in the previous question also. Transgenic mice are being used to test the safety of polio vaccine. This is also a true statement. Transgenic animals are made to carry genes which make them more sensitive to toxic substances than non-transgenic animals. This is also true that the transgenic animals are being used for various clinical trials. The transgenic cow Rosie could produce alpha lactalbumin. This is also a true statement. So here the correct option is C. The number of varieties of rice and basmati rice respectively found in India are, the correct option is C. The increased crop production during green revolution in India was mainly due to, it was mainly due to agrochemicals and better management practices. So here the correct answer is D. The Green Revolution increased the food supply to triple fold. Nematode specific genes were introduced into a tobacco hose plant by using the vector. So here the correct answer is agrobacterium. What was used to deliver the normal copy of gene in the gene therapy trials for ADA deficiency? It was a retrovirus. The process of RNAi involves, straight away the answer is silencing of a specific mRNA due to complementary double-stranded RNA molecule. Mark the incorrect match. All right. Gene therapy, use of retrovirus to introduce cDNA into the host cell. It is correct. PCR used to detect mutations in genes in suspected cancer patient. It is also correct. ELISA, detection of non-proteinaceous antigens. No, this is false. So the incorrect match is C. Next, GEAC stands for Genetic Engineering Approval Committee. Biopiracy is the exploration of natural resources. No. Exploitation of a region's biological resources or indigenous knowledge unethically and without providing fair compensation. Yes, it is the correct answer. India successfully protected itself against biopiracy in a case where in late 1997 the United States Patent and Trademark Office granted a patent to the basmati. If one needs to derive a cDNA of a human insulin, which cells should be taken up for extraction? Of course, beta cells of islets of Langerhans of the human pancreas. The mobile genetic element is transposon. Herbicide resistant gene in a plant is BT. Cotton plants can be protected from corn borer by the proteins encoded by cry-first AB. Cry-first AB resists to corn borers particularly. RNAi as a mechanism of cellular defense takes place in all eukaryotic organisms. So this was the discussion for the NEET mock test 10 of zoology. Thank you.